Okay. Good morning, everyone. Welcome to the CCEMP seminar. I think before the start, I would like to begin with acknowledging the voluntary people of the Korean nations as the traditional owner of the lands on which our Parkview campus stands. I also acknowledge the traditional custodians of various lands on which you are all joining me from today. I pay my respect to the elders, past, present, and emerging, and to the Aboriginal elders of other communities. Good morning, everyone. My name is Xing Hao. I'm a CCEMMP PhD student at the Monash node, and I am the acting chair of today's seminar. Today it is our honor to have Associate Professor Michelle Dunstone here to give us a 45 minutes talk. Michelle is a former ARC Future Fellow who has recently moved the teaching and research role at Monash. She obtained her honors degree in the biochemistry at Monash and her PhD in the lab of Professor Michael Parker at University of Melbourne. Her work focused on the MACPF class of pore-forming proteins of human immune system. And in 2007, she published the first representative structures of MACPF pore-forming proteins on Science, and followed by Cryo-EM structures of many different MACPF CTC proteins. Today, Michelle will give us a talk on the topic of discovering how pore-forming proteins evolve different assembly and targeting mechanisms. And just a reminder of all the audience, this seminar will be recorded. And if you have any questions during the talk, please type them on the chatting so that we can ask Michelle after she finishes her talk. So it's all over you, Michelle.
Okay, thank you. Yeah, right. Thank you so much for asking me to talk. I'm very, very nervous, I have to admit. It's been 2 years since I've given like a big full talk, so we'll see how we go. All right, so I'm absolutely fascinated by all pore-forming toxins, and majority of my work has been in one family of pore-forming toxins called the MACPF CDC toxins. So you'll hear me say MACPF and CDC a lot. And what I've done is I've used crystallography and Cryo-EM to take snapshots of different states of these pore-forming toxins or pore-forming proteins and actually try to put together the assembly pathways, which was really all very great and well until possibly about a year ago when I started to really look at some of the work by Till Bocking, and it started to open up the idea that, or confirm the idea that maybe these assembly pathways are garden pathways, because sometimes what you see is very selective. So I'll go through that. Okay, so I'm going to just get my thanks out of the way now, just because it's been such a big team effort. Thank you to Bradley Spicer, Charlie Bailey-Jones, Susu Payne, Stephanie Kondos, Ruby Lord, Tom Caradoc-Davies, all been really integral at Monash to getting the structures. Cyril Rabul for modelling and really kind of like the kind of nuts and bolts of how these proteins work. James Wistock, Helen Sable, Natasha, and another Natasha from Birkbeck. And another thanks to Till. I will mention some of his work just to kind of demonstrate that garden path idea. I do like Mr. Titan, so he's there. I do try to give him regular hugs, but I do actually also want to acknowledge little Miss Polara, who was there before Mr. Titan, because this is what this area had been built up and was like the Polara data, and maybe that kind of led to the garden parts, maybe not. So essentially, let's see if I can get this moving. So what we've found is that these proteins have really flat interfaces, and it's through these flat interfaces that they can oligomerize. and they want to oligomerize, but only on the right surface. So we're simulating that a flat surface here, that might be the cell membrane. And what we want to do is quickly go to the, so this, looking at this bit top down, is what we'd call the pre-pore. And then if we look sideways, we look at the molecular mechanism, and it basically punches down into the cell. So it takes these alpha helices with these cryptic transmembrane regions and punches into the cell. So this is the MACPF CDC dogma of how to form a pore. Okay, so how did we get to all this, and why is it important? Okay, so taking a step back, so that's kind of like the general mechanism of hole punching proteins. If you haven't seen them before, I hope that you're really excited by them, that the conformational changes are amazing. But that was just one family. There are many, many different families. And what we find is that, say, if bacteria wants to use hole punching proteins as one of their defense mechanisms or attack mechanisms, they'll actually have more than one family. So they have a lot of redundancy built in. The same with fungi. They use lots of different types of hole punching proteins to achieve an outcome. And the same goes for the human immune system. So we have many different hole punching proteins, including peptides. So this is not, when we try to use hole punching proteins, we use many different ones. It's not an isolated thing. Do they have a common mechanism? I'm going to keep it really simple here. The concept is that they're a soluble monomeric protein that's happy by itself in a folded state, that it changes into a ring-shaped oligomer, and that this leads to pore formation by a conformational change in the lipid membrane. Okay, so they're the three common things in pore formation, but there's a lot of varying themes, which is important to know, and that's really what this talk is about. And the biotechnology applications are really important as well. So we have Nanopore technology. So the classic of this is the generation sequencing. So you might know this as MinION by Oxford Nanopore technology. Now, so most of what they developed was actually developed on alpha hemolysin, which is a heptamer. Unfortunately, alpha hemolysin didn't have quite the right properties, so they had to move to another protein. I can't remember the full name. It's called APP. It's a monomer. So it has much better properties, but the whole back-end research and development that went into MinION was made on pore-forming proteins. And they are always, there's many different companies and researchers looking at pore-forming proteins to develop more nanotechnology. Okay, so we'll stop that there. And then the other aspect, main aspect of nanotechnology is their use in genetically modified crops. So you might have heard of BT crystals or BT cotton. In Australia, 90% of our cotton, or 90 to 95% of our cotton, actually is genetically modified and has 3 pore-forming proteins put into the genome, and it expresses in the leaves and the tissue, so such that when the moth larvae come along and eat it, that's not the right one. When moth larvae come along and eat it, then the moths die. So we do need to redevelop this technology because we are getting resistance in the moth populations to BT cotton, unfortunately, and there is an urgent need to develop new pesticides against not just moths, but mosquitoes, beetles, roundworms, and things like that. So that's really an important area. In the MACPF CDC field, there is one potential protein called GNIP, which has been published, and that helps protect crops against beetles. A third application is potentially immunotoxins, which I won't talk about here. It's pretty quiet, but yeah. So that's why I'm really passionate about the area, because it got such brilliant applications in nanotechnology and in as pesticides. So and I keep saying that there's different folds lead that can kind of basically lead to the same end outcome, which is a pore. So this is just one quick snapshot of a few different folds. We can classify them loosely into alpha pore forming toxins because they go across the membrane using amphipathic alpha helices. The other ones are the beta pore forming toxins, which go across the membrane using amphipathic beta barrels. So that's pretty simple. Lots of different families there. And this is just there are probably about 10 different alpha pore forming folds, and at least I think something like 20 for the betas. And then just to highlight the difference between the different beta pore forming barrels or pore forming proteins. So there are the small ones. So lysin family uses heptamers or nonomers, things like that. So they're quite small. Whereas the MACPF CDCs use giant beta barrels. So I just wanted to kind of pause and take check on this slide. You can see with the top-down view comparing, when you compare the giant beta barrels that you see with MACPFs and CDCs to the small ones from, say, the lysin family, because you've got so many more units, so much more can go wrong in the assembly pathway. Obviously, it's just some things are going to go wrong. So it's actually quite amazing that you can actually make these beta barrels in the first place. But as you'll see, right towards the end, I'll show you some of this EM where we're just basically chucking out some of this data. And so maybe that data is actually important in understanding the assembly pathway or off pathways. So going back to how MACPFs are big, one important thing to know is that they're so big that they can actually allow the transport of whole folded proteins through the barrel. And again, this is why this family is of interest for nanotechnology. We do have evidence that actual proteins do go through the barrels. So, for example, granzymes need to be transported by perforin barrels, perforin pores. And likewise for the MAC, lysozyme goes through the barrel, potentially also antibiotics. And I just want to know what all of these MACPF CDCs do in life, and there hasn't been any kingdoms or areas that I haven't found a MACPF CDC, and unfortunately we just really don't know what they do in many of them. We find them obviously in bacteria, malaria, Plasmodium, species that really rudimentary animals, so sponges and sea anemones. They're in plants. We know even less about the plant pore forming proteins. They're actually involved in development in insects and sea urchins. Again, there is research in that area that's slowly coming to some ideas, but why would you need a pore forming protein in development? It's an intriguing question. There's also obviously fungi I'm going to be talking about, and I'll be talking about proteins from macrophage and the immune system. So with the MACPF family, there's a common domain. So I've highlighted that in blue. So that's the domain that's involved in, well, is in charge of oligomerization and hole punching. So you saw that in the movie. So that's fairly straightforward. That's fine. Many of the proteins we look at, we know that they have domains that allow them to target specific proteins or lipids. And so that's another easy thing to get your head around, because how do we actually get these big chunky domains that are involved in making pores to the right part? Well, we just put on evolution came up with an answer. It just put on a domain that allows it to target specifically. In the case of the fungal toxin, it actually uses a second molecule, so it's a binary toxin. So then there's a third class of ancillary domain, and these are what I call the scaffold ones, because they don't actually touch down on the target surface, but they are involved in forming that really big barrel. So they're there to kind of connect to each other like Lego and form the barrel. And these domains become really obvious and important later on. So we find these domains in the membrane attack complex and in MPEG, which they're both immune toxins. Okay. So we're going to be looking at some snapshots, and before we do that, so the first snapshot obviously is to look at the crystal structure of the monomer. So I keep talking about this MACPF domain or MACPF CDC domain. So this is the common domain here. So this is the fungal toxin, has this really bent beta sheet in the middle. In red are the actual parts that punch down into the membrane. So they go from these alpha helices into these beta sheets. And we see the same fold in PFO, which was the first structure effectively for the family, and it came from Michael Parker's lab where I did my PhD. What he didn't know at the time, another project that he was working on back in the 1990s was actually related to CDCs. So he was for a little while working on perforin, as were many groups around the world. What no one knew is that these two actually related, and it took another protein that we were looking at to actually kind of connect the dots there. But as you can see, they all have this flat domain, which with the bent beta sheet holding up kind of the scrunched up transmembrane regions. Okay, so they're all shown in red. So the transmembrane regions, they only really need to have about 3 cryptic hydrophobic residues to span per strand to span across the membrane. So it's very hard to pick them just if you looked at a sequence. You do need to look at the structure. There is another area called the HTH region, which we think is important, although now suddenly if Gaz-Dermans are related, they don't even have an HTH region there. So it's kind of like a latch for the gate, but we don't really know. I think what's more interesting is not to look at where all the atoms are in these proteins, it's to try and connect the dots in how they assemble. And the first person to do that was Helen Sable from Birkbeck College, who did this fantastic job of observing both pre-pores and pores. I think she might have done this on the Polara. And so this is a demonstration of, I think this is about 30 angstrom resolutions. You still can get information at that resolution. That's important. So we know oligomerises on the membrane, they observed pre-pores. And again, matching that up with the structure, you can start to think about how those clusters of helices would form 2 beta hairpins. Before we had these big structures on the right, the EM structures, there was ideas of maybe the domain 4 region contributing to pore formation and things like that. So you do need to have all this information. This was also supported by a lot of biophysics from Rod Tweeton's lab, where they worked out that there were 2 beta hairpins per molecule that inserted into the membrane. So that was completely separate from the EM, but contributed. And these CDCs make these really big pores of up to 50 units in the rings. So that's like a 200 strand beta barrel. Okay. So we're just kind of gathering this information from static structures. And one thing that always resonated in my head when you speak to Helen about these structures is that when she observed the pore structures, they had a higher oligomer state than these pre-pore structures. So the pre-pore structures could have actually just been a dead end assembly path, dead end to the assembly pathway, because they didn't have enough units and they couldn't punch down. So it's so tempting to kind of go, oh, this is definitely the pre-pore, and then it punches down simultaneously. So I'm going to come back to that. This effectively did become the dogma for the field. Okay, that's very fine. We do get correlation of that kind of pre-pore punching down mechanism with chloratolysin. So this is the first MACPF protein that we're able to take. It had beautiful biochemistry properties. What we know is that it's expressed in the fruiting bodies of oyster mushrooms. You've probably actually eaten this toxin and it hasn't hurt you at all. We know it can kill Western corn rootworm and Colorado potato beetle larvae. Now, they might not mean anything to you. Gets me excited because these are really important for crops. Sorry, see if I can just take this back a bit. What we found is that we're able to do different snapshots of the assembly pathway. We had the crystal structures of these A dimers. We had crystal structures of B that comes down. And then we don't really have much evidence. We have some evidence of these little arcs, but not on lipid membranes. But we do find that we can have these trapped versions of the prepores. And we've got trapped versions at different states. So, well, that's all really great. It's starting to support what we see with CDCs. So maybe this is how all of the MACPF CDCs punch holes. Maybe not. Okay, so we published that, and that was done on the Polara. So there we go. So these static snapshots, it's all very well. So this is the crystal structure here. We've got the crystal structure here. That's fine. We can fit it into fairly low resolutions, about 20 angstrom resolution data of prepores that have been trapped. But we artificially trapped these prepores using disulfide bonds. So you're automatically skewing the pathway, the assembly pathways. And this has kind of really weighed on my mind a lot. You know, we're kind of like pushing the pore formation into a corner, and then we're taking the observations that we want and we just throw everything else out. So, I mean, this was really good at its time. I'm not complaining. What we can see is we can just see the trajectory of how things unfold or how things can unfold kind of at a bulk level. So this is real information. How we interpret it is of interest. Okay, so this was really cutting edge at its time. So please don't laugh, but these blobby maps were so exciting, okay? These were the bee's knees of EM maps at the time. Okay, I like to be quite rigorous, so I did try to validate everything using biophysics, different assays, and some binding studies. Unfortunately, everything we do is still ensemble method. And now that I'm starting to talk about pathways and off pathways and things like that, what would be really perfect is if you could validate everything with single molecule style assays. So bearing that in mind. So back to the dogma. So as you can see, with the work with CDCs from Helen Sable, Michael Parker and Rod Tweeton, and the work with pleurolysin from my lab, together with Helen. So we're kind of getting this idea that, yeah, these pores, they form pre-pores, they come along and they punch a big hole really dramatically. It's like, you know, very aggressive kind of hole punching mechanism. But a lot was weighing on my mind at the time because there are other toxins that just, we already knew that they didn't quite have the same mechanism. So as you can see, there's already variation in the MACPF mechanism. So one thing I didn't mention before is that the CDCs stand up really tall and have this punching down mechanism, whereas pleurolysin, it doesn't really have that. When you think about the evolution of these molecules, CDCs would have split off quite early from the common ancestor, and pleurolysin later on. So they're kind of, you know, evolving different mechanisms for different evolutionary needs. Okay, so we've got that has a drop mechanism that has, and it uses ancillary mechanism that has no drop. Perforin kind of looks a lot like CDCs and pleurolysin. We think that there's no drop, and you know, it kind of looked like it made a straightforward pore, but we don't really know, and that work's still ongoing. And then weighing on my mind, there was this other protein called MPEG, which we really didn't know much about, and MAC, which we actually knew could assemble in more of a sequential mechanism. So we couldn't really work that out. So I'll run through, so the evolution is that MPEG is found in sponge. So it's probably the common, and we don't see any of the other genes for the MAC or perforin in sponge. So we think that the MPEG is actually the granddaddy of the genes for all the immune toxins, not including Gasderm if you count Gasderm in this family. I'm just going to be talking about MPEG, MAC and perforin. So somewhere at the notochord stage, there was a lot of gene duplication that led to the MAC proteins. It led to perforin, and it led to more MPEG. So it's quite a conserved protein. Okay, so let's go through what we know. So in MPEG, we did a lot of characterization of this protein, and I have to thank Charlie and Susu for just a massive amount of work. So MPEG, it's actually doesn't get released from the cell, it gets released into the vesicle, so I guess equivalent of the outside, so secreted into the cells. But in macrophages, it's expressed constitutively, there's inducible expression. Whatever happens, in its full form, it is located either in the ER, the Golgi endosomes, or phagolysosomes. Okay. I might get that. If you aren't muted, just mute. Thanks. Okay, so it's intracellular, and it's different again in that it's got this type 1 transmembrane region. So it actually gets fixed inside the vesicles, but it still has the equivalent it's the equivalent of being on the outside. So this is quite different from everything else we'd studied. It has these MABP and L domains, which have nothing to do with targeting the protein. What we find is that it's a scaffold. Whatever happens, we think that it's to do with the immune system killing within macrophage and other phagocytic cells. This is the massive effort that Charlie did in his PhD together with Susu. It's just an incredible job. I can't do it justice. I encourage you to talk to Charlie about his work here. It's absolutely fascinating. It would be another talk in on itself. And again, I'm not going to do it justice, but what we're seeing here is what we were able to do is take the MPEG pre-pore and see what it looks like when we put it together with liposomes. And what we find, so in this particular case, we actually express MPEG without the transmembrane region. So this isn't the transmembrane anchor. This is actually part of the MABP domain that helps kind of sink its little claws into the liposome. So we don't quite know if MPEG actually gets cleaved away from its transmembrane region. I suspect not, but yeah. So to have a look at the structure, we didn't actually get any crystal structures. So everything we've been doing is EM, which was just a huge breakthrough. And it's this region here that just helps kind of keep touch with the vesicle. It could also be part of its mechanism of insertion, but in a kind of really indirect way. And I say indirect because so this is the membrane of the vesicle that it's in. It's probably got this transmembrane region here as well. It's got the MAPP that's just kind of touching the surface, and we think that the L domain is here as well, also touching the surface, but we can't observe that. And then, as I said before, we know what the MACPF domain looks like really well. And we know where the transmembrane regions normally sit. Once we know that, we can look at the sequence and go, yeah, it probably extends out this way and inserts into this membrane. But the big problem about this protein is that these transmembrane regions would punch way down over here. So it's going to be forming some kind of trans punching mechanism. So this picture just shows again that we've got this MACPF domain and it's inside a pre-pore structure, and it's kind of touching onto the the membrane inside the vacuoles of or the different part, or let's say the phagolysosome at this stage, and it's touching onto that cell membrane. But then if it punches, it's going to be punching in the opposite direction. So we do see some evidence, some very light evidence of this. This is very low resolution structure of a transpore. It wasn't high enough for us to be able to definitely say that this was a transpore, but another group did follow up on similar research. So how we think it works is that on the inside of the phagolysosome in macrophage, you have this pre-pore that it's kind of embedded and making sure it's still got its little fingers into the membrane. This helps by preventing autolysis, because you don't want to unleash a pore-forming protein onto its own cell membranes. The scaffolds in the domains help form this stable pre-pore. It's very stable pre-pore, and it's controlled by pH. We do know that its activity is dependent on low pH. So then that spatio-temporal aspect where pore formation is controlled within the cell at a particular time. And then we think that it could target the actual, say, a bacteria that had been engulfed or things like that. Also, cells that are undergoing apoptosis are also engulfed. So it could be any number of things. And the thing that I wanted to point out, coming back to that, that the dogma that I had been showing for CDCs and paratolysin, where they have these receptor binding domains and they land down on the cell. When we model this system, we don't have any domains that are there for targeting the cell surface. And so it really confuses us how this can occur. But at the same time, it could be an explanation, because MPEG is there to punch holes in anything that comes into the vacuole. So maybe it doesn't need anything to, directly in its system to actually punch a hole in the membrane. One thing that's very, very speculative, okay, so there's really no evidence for this, but what you could speculate is that it's actually using other receptors. So similar to an immune synapse, where you've got a lot of different receptors sucking two cells together, and kind of really bringing the two membranes close together, and then you're getting perforin occurring. We could be having something similar happening here, particularly because the types of the bacteria that get engulfed into macrophage are just absolutely covered like a carpet with complement proteins from the immune system. So yeah, so could this be another synapse? I'm just throwing that idea out there. What I wanted to highlight is just coming back to this concept that you don't need to have a dedicated domain that is a receptor that is there to bind specifically to the target. And that's the advantage for the immune system, because if it did rely on that, human evolution or mammal evolution can't outpace bacterial evolution. So the immune system has come up with lots of ways of working out how to target things non-specifically. Okay, so we're still coming back to the animal immune system. So we've worked out more about the sponge, so the MPEG, which comes from as early as sponge through to almost every organism. There's some worms that don't have MPEG, but yeah, most animals do. So we've discussed MPEG. There's also perforin, kind of alluded to, but then in the back of my mind, and coming back to that assembly idea, there's always been the membrane attack complex, which we've known from the get-go has just had a completely different assembly pathway. But we know that they're all related, and we know that there was divergence. There's a common gene here. So MPEG, there was duplication. There's a common gene between perforin and MAC. perforin still probably keeps that simultaneous punching down mechanism, but then the MAC comes along with completely different mechanism. And it's worth kind of going through this in detail. So what is the membrane attack complex? Well, it's another hole punching protein that all the components are floating around in your blood right at the moment, waiting for the right opportunity to punch a hole. So predominantly it targets gram-negative bacteria. It can actually span the LPS layer and the outer membrane. So there's some suggestion that LPS is important for the MAC assembly, but we're not really quite sure about that. More about that later. It doesn't really need to directly bind to lipid membranes. We know that from a number of studies. Okay. So if we take kind of a cross section, what we're doing is we're getting this really complex pore. So all you need to know is you're starting off with this big blob called C5b. It doesn't have a MACPF domain in it, but it does grab C6, which does have a MACPF domain, and it gets C6 into the right shape. And it's only when C6 is in the right shape that you can get C7 to bind. And then when C7 binds, you get weak anchoring. And then when C8 binds, it's this complex MACPF domain. Well, there's two MACPF domains here. They come along, and then there's stronger anchoring and potential leakage of the membrane. And then you get multiple units of C9 come along. So already you can see that this has got a completely different assembly pathway. And so even from the early studies of MACPFs, the MAC has always been really annoying to me because it seems to be completely different from the other molecules or other members of the family. Or is it? I mean, maybe it's actually kind of just shades of grey. So the MAC is quite extreme. Another way of showing it is here. So remember, it takes a single copy of C5b, and then it grabs its C6, C7 comes along, C8, and then C9, okay? And it forms this big barrel. And we know this from the structure of the whole MAC, fantastic structure from Dorian Bubeck's lab in, I think she's in Imperial now. So she did some fantastic work on this MAC structure. But what we didn't understand is what actually controls that assembly pathway. So if we kind of just go back a bit here, I should say that there was the idea that C6, then the C7, then C8 comes along in a sequential manner, led to the idea that C9 is also sequentially added. Though we weren't really quite sure if maybe it was sequentially added and then it punched down, because, you know, this C9 barrel bit looks a little bit more like it could function like CDCs, but we really weren't sure. Okay, so we had the structure of C6, we did a model of C9, and again, it's got no domains there that could actually specifically bind directly onto a membrane, and nor should it. Maybe that's the whole point of how it evolved. What we think is that you get massive amount of complement activity on, say, the target bacteria. There's so much of it that just a little bit of the MAC that it can assemble will do its job. And I'll talk about that at the end. Okay, so again, this lunar landing mechanism of CDCs doesn't really fit with what we know about the MAC because it just doesn't have it. So let's chuck that idea out the window. How is the MAC able to assemble? Now that we've actually got the crystal structures, we can actually work that out. How is it controlled was the more important question. And is that important for function? I think I've really given the answer to that, because the immune system needs to be able to target anything and everything. So we needed to get some structures. So we got the before structure of C9. So C9 will be the important part here because we just didn't know what it was doing. We had some idea of the early stages of the MAC, but we just didn't know what its assembly pathway was. And in order to get really high resolution, we have this kind of cheap molecule called poly-C9, where it's just a homogeneous ring of C9. And we can use that to study at the atomic level the whole membrane attack complex. Okay, so this is the early stuff that we did. It's not the best, but we've got much better than now. And this is the crystal structure of the mouse monomeric C9. So these two structures really made a difference. So that's a better slide there. So as you can see, what we can do is we can look at the before shape of C9 and the after shape of C9, and actually you can deduce some really important things about its assembly pathway. Okay, so there were some other structures of the other membrane attack complex components, C6, C8 beta. We don't have one for C7, but there's several structures of C8 beta. And all the structures, when you look at that TMH1 region, so the first beta hairpin that inserts, it will be on this side here. But then when you compare it to the structure of C9 that we got, it actually went all the way through the domain and popped out the other side. It has this massive glycan on it. So it's big. This is a very hypervariable region when you look at all the different C9s across mammals. And yeah, so I should say mammals, animals, vertebrates, I should say. So basically, in all the C9s, we now predict that it will be this big chunky thing on the other side. So how's this important? Well, it's important because we know assembly goes in a certain direction. So this is the face that will bind onto the growing pore, and this is the elongation face that will take or accept the next molecule. And in all the structures, even CDCs and things like that, this face isn't really obscured by anything bulky. This is the only structure we know of which has got this big bulky thing. So this bulky thing has to move before the next molecule can come along. So as I said, we did a bit of an analysis, and this TMH1 is always on this side except for C9 where it's on this side. So we thought, well, maybe this is part of controlling the assembly pathway. If you look at it from the top down, assembly always goes in that direction. We know that because from the MAC structure. So if you want the next molecule to come along, you have to actually move that green section, so the TMH1. If the TMH1 moves, at the time we postulated that if the TMH1 moves, it has to insert into the target membrane. So that green TMH1 region is inserting into the membrane, and then that allows the next molecule to come along. And again, to really validate that, that observation, what we did is we put a disulfide bond. So we trapped that TMH1 region, so it couldn't come through the fold and it couldn't punch into the membrane. So we've got it trapped there, and then we tested its assay, tested it in an assay. So in this particular assay, if you have no C9 present, you're not going to get any change in turbidity, which was our indication of pore formation. If you add the wild type C9, you do get pore formation and you get this turbidity change. If you add the disulfide locked C9, it's completely dead. It's as if you've got no C9 there. And then if you add DTT, you can rescue that effect and it will insert again. So that's all very good. So this video is not the best of videos, but it gives you the idea that basically C6, C7, C8 comes along, you get some insertion here, and you can get some leakage from membranes, and then you get C9 and then insertion, so you have to get insertion and then binding. So already you can see that is just completely different to this big kind of hole punching mechanism. But they've got the same fold. So that whole dogma of like the fold dictates the activity, you can still get different pathways, even though you've got the same fold. And I don't know if that, you could get really hung up and think, all right, you know, how can you have the same fold that in some, they all hang up tight and then they punch down together, whereas in another one, it will sequentially insert, but maybe it's all shades of gray anyway. All right. Okay, so really what we've been looking at and studying using Cryo-EM is kind of almost like the different ends of the spectrum of assembly pathways and what's possible. And then taking a step back, remember that there's a lot of different MACPF CDCs we just don't know about. So I do feel that now the dogma for some of the CDC work has become, you know, it needs to be readdressed, and it's more like a garden path than an assembly pathway. Maybe not. And it's better to think about things in a really holistic way. So I love speaking to this audience because I know you're all into Cryo-EM, but I'm starting to think that Cryo-EM is not the way to look at assembly pathways, or not the only way. And the reason for this is because we know from AFM that we can see arcs. So having single molecule time resolved experiments to marry into the EM is really important. But for a long time, people who are experts, who were into the EM of CDCs, were actually opposed to the people who were looking at arcs by AFM or other methods, or by EM. You know, so there was actually at one conference there was almost a bit of a biff up about if arcs were real or not. I mean, you see arcs in our EM images and we throw them out. So, you know, are they real? The MAC actually has two different types of split washer assemblies, and even as far back as 2000, we actually saw these soluble spirals of CDCs. So, and this brings me back also to the MAC. So the MAC actually deliberately has all these off pathways. So it actually has these assembly pathways that are soluble and don't insert, but then they get kind of taken away by clusterin and vitronectin. The way the MAC, so possibly only maybe 1 in 1,000 MAC assemblies leads to a full pore that has an effect on the cell, but that doesn't matter. That's enough for the immune system to work. And it's basically these other pathways are taken into account. We do see this, and even for pleuratolysine, we see evidence of arcs. So we can kind of get an idea of how they might assemble, but we don't know if there's release of the transmembrane regions or not. Likewise, we see partial polyC9 formation. So it's important to actually take this into account and not throw out this information. The way I want to work forward on this, so this is an example from Michael Parker's PFO project that, so Till Bocking took PFO that was from Michael Parker's lab and started to study it. Basically, what you can do is at the single molecule level, look at liposomes that contain dye, and then marry that with how many PFOs assemble on that liposome. So this is an example of a liposome here that was accumulating lots of PFO. So then you can actually look at both signals. So you're looking at the loss of the dye from the liposome, but you're also looking at the gain of signal, and you're looking at that over time, and it's single molecule. Okay, so this is definitely something that we should be looking at doing more in the MACPF CDC family, and that's exactly what I'm going to be doing with the MAC. So what Till found, though, was that once you get that dye release, once you get that dye release, you're going to see 2 blue dots on the screen. Oh, okay. Once you get that dye, yep, I'm finishing. Last slide. Once you get that dye release, you can actually still continue get assembly of the CDC pore. So this is kind of counterintuitive and what no one else has really considered before. So what we want to do is actually, I'll just go to the last slide, is actually consider that as an option for the membrane attack complex, and that there are different pathways that we need to explore. Okay, I'll finish up there. It's important because, all right, I'll just go to my last slide. It is all really important because we want to know how to make this nanotechnology better. And we also want to know how to outfox things like the muff larvae, make sure that we can develop pore-forming proteins that these larvae can't actually develop a resistance to. So I'll just leave everything there on my acknowledgement slides. And sorry, I went over time. I forgot.
Sorry. It's my bad to disrupt, but it's really an interesting talk on the complicated pore-forming MAC protein structures. And thank you very much, Michelle. So if any question you have, please hand up your hand. Okay, we saw a few questions. So Patrick, maybe you can unmute and then ask your question.
Thanks, Michelle. Really interesting talk. I might have missed it, but what is determining the pore size? Is it the angle, the assembly angle, and is the curvature of the membrane contributing to whether you get completion of a pore versus an arc? And
it's the flat. It's definitely the interface. So different. It's the combination of the MACPF CDC interface and any of the ancillary domains around it. So for the pleurolysin, it's got ancillary domains which actually have these wings that kind of push it, so it becomes only a 13-mer, whereas CDCs have just a really, really flat face, and they don't have any other domains that are interfering with that oligomer interface. So they have, because it's flatter, they have bigger rings. It's unlikely that the curvature of the lipids would have that much effect because lipids are lipids and they're quite fluid. And it's important for us to remember that the protein structure will be much more rigid and it will move the lipids to where it needs the lipids to be, which is what we see when in the change of from pre-pore to pore. The lipids effectively move.
So what do you think limits arcs versus closed pores?
So arcs were do you mean like the small pre-pores or the arcs?
The arcs just look like that you've partially assembled and then it's stopped and hasn't continued the assembly to completion.
So the factors are more likely to be concentration and temperature. It depends on what you're looking at. Definitely for CDCs, they're very temperature dependent, although they tend to still eventually get to making a pre-pore. Sometimes just batch to batch variation can have an effect as well. So we don't really know. It will be different grids on different days. You'll see different proportions of pores.
But you're also seeing them in cells, if I understand correctly, in EM images of cells. So it's happening.
We've only really done, that's a really good point. So, and it's a point I wanted to raise at the end, that we've only really been able to visualize this on liposomes.
Mm-hmm.
The next step that Till and I are planning to do is to, Till has actually done some work on CDCs looking at virus-like particles. So let's say a step up from liposomes, but I don't think we've been able to really look at what happens on a cell surface using structural techniques. However, what you could do is really develop turf to be able to maybe tackle that question, which is exactly what we should be doing, because we could be looking at all these fancy things under EM, but it's not what's actually delivering the death blow on a cell.
Thanks, Michelle.
Okay, we still got a few questions, so we are a little bit run over time. Are you happy to stay over
that's
for
fine.
a few minutes, Michelle?
Yep.
Cool. So maybe next question, Hai Tian. Can you unmute yourself?
Yep. Great talk, Michelle. So you have mentioned earlier saying that the pore formation is triggered by pH. So is the oligomerization of the single subunit also triggered by pH?
So only certain CDCs and MPEG is triggered by pH. And with MPEG, we only really ever see it we've only observed it under EM in a pre-pore. It really prefers to be in a pre-pore state. So we wouldn't really be able to decipher that. And everything else that we've done has been ensemble methods. So, it would be a really good question to be able to say, would a single MPEG be able to change shape in acidic conditions? MPEG really likes to hang out with each other. They all hang out with each other in a ring. So it would be incredibly hard to observe. It's a good question though.
Right, yeah. So you purified everything like in a pre-pore assembly instead of single subunit assembly.
So this is the back end. What really happens in the lab answer is that you can see a peak that might correspond to a monomer for MPEG, but you also always see something coming out in the void volume of the size exclusion column. And I kept seeing Susu show these size exclusion chromatography pictures, and I'm like, I think you need to just shove that void volume sample under EM, just have a look at it. And I just recommend everyone does this, like for your void volume, just have a look at it under EM, because you never know what you'll discover. So for years, Susu was trying to just crystallize the monomer peak from size exclusion, and she just gave some sample to Brad. He looked at it next time he was under EM, and we saw these most beautiful donuts. So she had effectively been throwing out donuts for years. So, and that's the preference that molecule wants to be in.
Okay, cool. All right, thanks.
Cool. We still have a few questions in the chat. So Isabella Rulia, could you unmute yourself and ask your question? Or
hi, Michelle.
Hi.
Beautiful talk. Fascinating story you're telling here on I like the journey. I guess my question regards the work from Charlie, where he typically observed 2 stack rings, and that's the majority of the particles. If I looked at the diagram you show, do you think that means anything, or what's the
totally
of that?
Yes. Protein is always telling you something, right? It was one of the biggest clues for me, and I had to have lots of hand waving moments about this. What I didn't say is that those donuts always liked to be stuck to each other, and it was as if they were trying to shield their residues from the water, right? And so from crystallography, you know that if something wants to crystallize, it's because it prefers to be in that state rather than having all that water around it. And I thought maybe that's that surface prefers to interact with the lipid membrane. So it took a while, but we got to the point where we basically put the rings in the presence of liposomes, and so then they must have been in an equilibrium coming on and off, so that the 2 rings coming on and off, so then they can sample the liposome surface, and then once they sampled the liposome surface, they preferred to be on the liposome surface. So then once you add liposomes, the rings preferred to be on the surface of the liposome. And then the debate was, well, which way were the rings? Because we didn't have the resolution to work that out at that stage. And that was my other hand waving moment, was that I had an idea that they might be upside down, but we had to get the data to do that. So yeah, the rings were telling us something as well. Always think your proteins are beautiful. They're always telling you something.
Thank you.
Ooh, as the time has approached 11, I will just wrap up a little bit earlier before letting Josh to ask his question. So thank you everyone to join us to the CCEMP seminar. And we really enjoyed the talk of Michelle, and hopefully can see you from the next seminar next month, and you can find out the information on our CCEMP website. So for those is interested in the following questions, please stay for a few minutes, and thank you everyone again. So
yeah, so I can answer Josh's question.
Yeah.
Do you mean inhibitors of pore forming proteins, Josh?
So yeah, I was just thinking about, you're describing this mechanism, and it's an attack, and I guess you talked about how they're a bit agnostic to the membranes they're targeting. Some of them have evolved mechanisms for just hitting any membrane that happens to be in proximity, right?
Yes. Yeah.
Have you seen, bacteria, pests evolving defense mechanisms against these pore forming toxins? What's
no, yeah,
they evolved to dodge this?
There are just not many definitive inhibitors. The ones that, so the membrane attack complex does attract inhibitors. It's the only one, and it's the only one that has this sequential assembly pathway, definite sequential pathway. And so that might be the Achilles heel of the MAC system of assembling. It's much slower. There's time, there's exposure of sites that you could inhibit. So again, that's where if I can use more EM and more turf to actually look at the kinetics of every single pathway, you could actually then start to design inhibitors of the MAC, which would help tone down inflammatory conditions. But when you look at the ones that kind of point towards more of a simultaneous punching down mechanism, they don't really have inhibitors. Like, how would you inhibit it? It's an interesting question.
Yeah, it's just at the beginning you mentioned resistance from pests towards this. Yeah.
Yes, so that's because they change the receptor that they bind to.
Right, yeah.
So you're not looking at inhibiting the action of the pore forming domain region, you're looking at how it recognizes the surfaces. So these companies go to a huge effort to look at different variations of their toxins to try and create new genetically modified organisms that overcome this. And even our own CSIRO, they have a pest management program that involves having fields which are not GM, so that you have a reservoir of insects that still can be killed by the GM, by the toxins. So it's very complex, but money is spent on this problem, and the problem is based on the domain that targets the proteinaceous receptor.
Okay, yeah, that makes sense. Thanks.
Yeah. So one thing I didn't mention at the end, I ran out of time, is I'm like, well, I don't think the MACPF CDC family will actually cut the mustard when it comes to developing pesticides. What you need to do is look for pore forming proteins where you can easily engineer its targeting domain. So on that note, I'm kind of switching my work to toxin 10 family. and how they work, because you can actually, like beads on a string, you can take the receptor off the domains off and put a different domain on.
Yeah.
And they're much nicer toxins to work for.